Alrighty, everyone, and we're just about ready to get started. So, hello, everyone. Welcome to the Biotechnology Workshop with Mr. Sajid Hai. Sajid, we're so glad to have you here.
Thank you so much. Hi, my name is Sajid. Just a little bit about me. I am 27 years old. I've actually kind of grew up in the Bay Area, in Cupertino, California specifically. I've lived here majority of my life, and I've always had a really, high interest in specifically microbiology. Now, does anyone here know what microbiology is, or specifically what that field, is concentrated in specifically? I see a lot of nods. All right. Well, microbiology is the study of microorganisms, specifically study of bacteria and viruses. So it actually it has a lot of overlap with pathology, as well as a lot of detailed a lot of detailed projects in study of bacteria. So in a way, I had a lot of understanding of what this current pandemic was already coming into it, because we did a lot of research in I don't know if anyone is familiar with Bacillus anthracis, anthrax. You guys know what anthrax? I've heard of No one has heard of anthrax. So anthrax is actually is a very dangerous bacteria that's used in biochemical warfare in the past. I don't know if anyone's familiar with like the letter openings, where they used to they actually stopped licking the letters in the post office because there was actually a horrible issue where someone tried to kill off people, because anthrax is actually spread in the air. So very similar to, although not different than COVID, because COVID is a virus. This is actually a bacteria. And the way that anthrax works is it actually stays dormant for years. And the only way that it actually kind of survives and lives is only on human contact. So you can imagine how crazy this got when the second, it got into the air, people breathed it in. And what it does is it actually causes you to lose pulmonary function. So you start having difficulty breathing, and you can die. So this is the research I did in my undergraduate as well as graduate studies. So it kind of helped me understand of the things I really loved. And I just love the study of bacteria. If we think about it, right, I don't know if anyone knew this, but we actually our bacteria cells in our human body outweigh or outnumber our human cells 10-1, which is crazy to think about, right? So for every one human cell, you have 10 bacteria on you. Think about that, which is crazy, because that means it's a part of you, right? It's a part of your human body, your biome, as they call it. So what I do right now is I actually work at Thermo Fisher Scientific, which is a biotech company. Currently, what they are doing is they're one of the leading companies in drugs of abuse testing, calibrations, as well as right now, we've kind of channeled all of our resources into fighting, as well as specifically countering the pandemic currently. We currently manufacture the TAC-path RNA PCR control, which is utilized in testing, obviously, for COVID. And the control is it's a negative control, right? You basically test whatever sample is against it. And if it matches up in terms of, negativity or positive, as if you guys are familiar with how you read a gel, when you test the gel after you take the samples, you have to read it based off the proteins, right? And how far the segments are read out. But it's really cool stuff that we kind of allocated all of our resources into. So right now we're doing a lot of work into specifically manufacturing the control for COVID, as well as we're doing a lot of vaccine development manufacturing. So right now, I don't know if anyone's familiar with how the vaccines are currently being distributed, but there was two hurdles for the vaccine specifically, which took it a little bit longer for it actually, specifically the Pfizer vaccine, for it to be manufactured. So one is delivery method, because as I don't know if you guys are all familiar with, how RNA, exterior RNA is acknowledged in the cell, right? If there is any sort of foreign piece of DNA or RNA floating around, we all know what happens is the body actually just destroys it automatically, right? Because it acknowledges it as a foreign entity, right? And it's not not like cells. So what happens is the way that we kind of tackle that issue is what they created was a small lipid nanoparticle bilayer to protect the mRNA. Now, this vaccine is different in that previous vaccines utilized parts of the dead virus or parts of the dead bacteria itself. But this was a whole new venture, because what they wanted to create was they wanted to amplify the production of not only the antibodies and the antigens, right, inside of the cell, so that you didn't need, right, you have that amplified response when that COVID virus came into your body, right? So the way that they counteracted that measure was they used a lipid nanoparticle layer to ensure that the mRNA would actually be able to be enveloped into the cell. And then the second hurdle was obviously transportation, right? I don't know if anyone has been kind of paying close attention to the news, but the biggest issue is obviously transportation in terms of these vaccines have to be at a specific temperature, right? They have to be transported around 2 to 8, and actually negative or almost negative 20 degrees Celsius, as a matter of fact. And it has to be kept at these conditions for an extensive amount of time, right? So these are huge hurdles, because normal warehouses don't hold dry ice or liquid nitrogen, the things you need to keep these at stored negative 20 degrees. So a lot of these warehouses are Thermo Fisher warehouses all across. I'm pretty sure you guys are familiar with Project Warp Speed. There's a lot of warehouses all across the United States, which we help manufacture these dry ice and help store the vaccines all across the nation. But that's just a little bit of what I do. I personally love working in the lab. We actually come in shifts to help fill the controls. So currently what they have us doing is they have us kind of coming on off days. So one day we come and fill the specific, specific requirements for the TAC path controls for the RNA PCR test. So what that looks like is, I'm sure you guys are familiar with pipetting. So what they have us do is they'll have us spread out on workbenches, and what they do is they have us actually sign up online so that they can monitor who's signing up on which day. What that allows for contact tracing in case if anyone tests positive. Mainly because since we're manufacturing, which is a huge manufacturing plant, we're one of the first providers of controls all across all across the nation for hospitals, for clinics, right? Whenever you're taking your at-home test, what the control that we're providing is we're actually sending those to the lab so that they can test your sample against it, right? So we're creating millions of these a week. So we have to be able to ensure that we're doing this safely, on time, as well as we're ensuring that if there's any sort of individual who has tested positive, we can make sure that we have traced down all of the individuals they've been in contact with as well. So it's a lot of work, but honestly, during this pandemic, it's shown me how much the biotech world has evolved. Not only in terms of vaccine development, not in terms of therapeutics and drugs of abuse, development, but now we're going so much in terms of we're going above and beyond just reactionary medicine, right? We're trying to go and be more proactive with our therapeutics, and I think that's a bigger jump, because obviously with the next pandemic we don't know what exactly could happen. So I think with the new vaccine development, it'll allow us to create these vaccines at a faster rate. But if anyone has any questions right now, I'm letting the floor open. I know I talked for quite a long time. I get a little bit how do I say anxious. Whenever I'm outside of the lab, I love talking about what I do. So
All right, awesome. So me and my partner actually have some questions already. So I guess we'll start off with a few of those, and then we can create some sort of discussion going so that the audience can also jump in with their own questions. So you actually answered a lot of our questions with your speech. That was really amazing. But one of the things we were wondering is, so we had a biochemistry speaker yesterday, and she said that her company uses a lot of Thermo Fisher products. So could you kind of explain like your thought process on how you create these products and how exactly would you sell it to other companies?
absolutely. So yeah, we sell a lot of different products all across the board, from organic chemistry to antibodies to particles. So the specific, do you remember what specific product, Rishit, that they were utilizing from our company that they?
She did not actually mention, but she says that like a lot of her company in general just uses a lot of your guys' products, and she said that they're really
Absolutely,
helpful.
yeah, yeah. No, we do. So it's cool because Thermo Fisher is one of the largest biotech companies in the world. In terms of biotech sales, I think we probably are number one, number two, I believe. We just because we don't only create the the testing reagents, controls, calibrators, right? And calibrations. We create the instruments themselves, right? So we go so far as to not only create the testing material. We actually create latex gloves. We manufacture a lot of polystyrene, which is used in plastics for anything you could possibly imagine. The squirt bottles you see in your lab. We actually manufacture those as well. Nalgene. I'm pretty sure if you guys have any Nalgene products, we actually partner with Nalgene to create a lot of their plastics, just because the durability of those plastics are a lot better than most, if you actually think about it. We create not only just instruments, but if you guys are familiar with like mass spectrometers or proton NMR, those are large machines, multi-million-dollar machines that actually break down the composition of elements, and they give you like a breakdown, right? Like mass spectrometers, for instance, what they'll do is they'll give you like, okay, you're trying to study a molecule of, I don't know, let's see, like a methanol, right? For instance, and then what it will do is it'll actually tell you the breakup of the carbon, right? The hydrogen, whatever else is part of it. So pretty cool stuff.
Awesome. Right. So after your talk and the way you answered that question, I think everyone can see that you really love your job. So could you kind of say what inspired you to become a biotechnician?
Oh, that's a good question, man. What inspired me? Oh man. We are mass producing items. Yes, we are manufacturing. So initially, Thermo Fisher is not just one company. There's a lot of entities that they have bought now at this point. Fisher Scientific is one of them. Life Technologies is another. Invitogen, if you've heard of Invitogen Biosciences, they are also a part of them as well. TagPath is another one. So there's a lot of like smaller companies that have been acquired, if that makes sense. Microgenics Corporation is actually the technical name of the company that I'm a part of. They used to be a part of what they called Ceradyne and Duke Standards. So with that's the old name for like the plastics and it's very technical, but they they've kind of grown, if that makes sense. But for me, like, I just love biology. Like, I love how life works. I love seeing the process of even understanding how you eat food, like just how simple that is. Like, now I can tell you, like, I'm eating this, this little bit of naan, right? And there's some roti and whatever else, and aloo, right? I know exactly how it's breaking down. Like, it's crazy. Because if you think about it, right, the starch and how the carbohydrates, right? The first thing that is being broken down is in your tongue, right? Because your amylase, right? Your amylase is automatically breaking down the carbohydrates. And just how the entire breakdown all throughout your esophagus, it's just incredible, because there's a process at every point, and how even the energy is never wasted. To me, that blows my mind. And then the second thing that made me really really fall in love with biotechnology, and just biology in general, was I had a marine biology class that I absolutely loved. And for me, like, marine biology, although it's not as, it's on the microscopic level, it's incredible, because you get a full appreciation of not just what's in the oceans, but you get an understanding of moon cycles, right? The tides, how that's related, king tides, the best time to go surfing, even, right? Like, there's so many things that come from that. Even just understanding the different currents that are a part of how unique the California bight is, for instance, right? And the California coast, right? We have giant kelp, which is one of the fastest growing organisms in the world. And just understanding that is crazy. And we have these sea otters, who are these incredible protectors, right? They're the guardians of the kelp forest. Just small things like that really made me love the ocean and made me love biology. I think from there, it kind of gradually made my love more. And I think my professors as well. My microbiology professor was my favorite.
All right. So building off what you just said, it seems like there's a lot of curiosity involved in your field. Are there any other like character traits which a person who wants to be in your field as well should have to be successful?
Honestly, like, you all are so young, man. Like, just for me, I'm just curious. So I love learning more. I love understanding and just trying to pick the brains of the most intelligent human beings in the room. So for me, whenever I go to even meetings, right? Even in my work, I know for a fact I barely know anything, right? So what helps me is kind of following around and mirroring these incredibly minded metrologists who have their PhDs from Harvard, right? Because what helps me understand is their minds have just been carved through years and years of challenges. So what the little bit of insight that I can get from the moments of interaction helps, because then I can see their thought process, right? Because I'm like, oh man, these guys they're just they're on top of their toes constantly. And just seeing those interactions, it makes for me, it's even better than reading chess strategy books, like, for instance. Like it gets me inside of their minds, and that's the best. Like, they're the smartest human beings in our field for sure.
Right. So you also mentioned that your professors really got you interested into microbiology as well. And I'm sure that there's a lot of people in our audience who kind of want to find some way to get into microbiology or biotechnology. But like, since school's online and stuff, you can't really get inspired as easily. So do you know any opportunities, whether it's online or after COVID's over, or anything which our audience can take advantage of to kind of get a firmer grasp on biotechnology?
Absolutely. Yeah. There's a lot of opportunities. I know even through our company, we've done a lot of work with the Tech Museum in San Jose, as well as a couple of other places and organizations in the community. However, I think it's, yeah, as you mentioned, it's really hard to be as interactive, especially with biotechnology, in a safe manner. But I will definitely compile a list of resources, because there's a lot out there for sure, in terms of STEM education. And I have a lot of really, really cool and interesting work in, like, Alzheimer's, for instance. One of my friends, he started up a company in Mountain View, for instance. They do incredible work with actually utilizing genetic profiling and actually databasing for Alzheimer's specific diseases. And it's incredible the amount of research and work that he's doing on the ground. So like things like that, where there's more opportunities for like startups and you can kind of mirror and understand what they're doing. I think those opportunities might be even more so advantageous, for instance. So maybe I can get you guys in contact with like new, startups who are trying to get on the ground or doing some really, really fascinating gripping work, where you maybe can intern, for instance, or possibility wise, like, summer volunteers, opportunities, I think would be great, because I think especially in this pandemic, it's hard to get those opportunities, like you said. But I think just getting the experience now, it's huge.
Awesome. Okay, so someone in the chat actually had a pretty good question. So in your job, do you work with a group or do you often work alone?
So, no, that's a good question. I work in conglomeration with a lot of groups. It's kind of weird. Like, I float around with like purchasing. I help out sometimes. I help out with research and development. I have like projects with the particles group, for instance, in antibodies. I'm all over the place, to be honest, because when I originally started, I was mostly doing purchasing, specifically the raw manufacturing of material to help make everything we have in-house, which was a little bit more of a finance role. And then I kind of used that to pivot into the lab role, which was kind of cool, because then I kind of understood the front end of it, right? Oh, why are we buying blood, right? Why are we buying human plasma? And then kind of understanding, okay, we're trying to maximize our profits to ensure that, okay, we're, the revenue margins are adding up, so that whatever we're making in-house, that we're maximizing the percent yields of what we're creating, right? So then when I got to the lab, it made sense. I'm like, yo, I can't be wasting this product, right? I just maximize the amount of revenue that we've created. So me utilizing this product to its, especially this, like, even if it's like ethanol, methanol, very simple things, I understood the value that it had, right? The value that it added to the company. So it was cool. I think getting a different understanding of these different roles in the company. But the group that I actually report to is the group that does the specifically logistics on ground. My manager, give me 1 second. Yeah, so basically I am reporting to the man who just literally, he's in charge of everything on the ground of the site. So I really, like, it's kind of weird at this point. I have no group that I'm part of, but I'm a part of a little bit of research and development, as well as with the metrologist. I do work with biochemists, but I work mostly with metrologists. Metrologists are they're kind of like an in-between between like genetic engineering and yeah, because they do a lot
Okay.
more like particle work. Very cool stuff. I can go into more detail about that later.
All right. So another question from the chat is, do you need a degree or specific course in college to work in biotechnology?
Yes. So for biotechnology, I would highly recommend. First off, you could always do. I would definitely try to specialize in maybe like biology specific, microbiology, molecular biology, whatever fascinates you the most. For me, I loved microbiology and biochemistry the most. So those are the two areas that I really wanted to concentrate on. So I would recommend that for undergrad, but I would go so far as to say you definitely need a graduate degree. Maybe even you don't need a PhD. However, sometimes in the field, they do look at PhDs a little bit differently, especially if you're doing more research and development. They look at doctorates, especially with because that means you've done your thesis, you've done your extenuous amounts of research in terms of whatever um, field that you are looking into progressing, right? Because with the PhD itself, you're putting yourself up for publication as well. But I think honestly, you can do it with a graduate. You can do it with an undergrad degree in bio and microbiology, molecular biology. If you want, you can get a graduate degree as well. In some people, what they do is they get a business degree in biotech to kind of get that a little bit of mixture of the biology with the business. But it's up to you. And they do pay. They pay a decent amount of money. I don't know what you're looking at for money wise, but I think they do a decent job paying me.
Okay, great. So we're gonna bring this back to your professional life. So is there ever was there ever a single moment which was like how do I explain this? Was there ever a moment that kind of like stood out to you that was like really interesting or like out of the blue in your work day? And could you like elaborate on that?
Absolutely, yeah. I guess like the craziest part was just seeing the impact that the work that I do has for not only the health field, but just completely, breaking down walls for different ways of testing and countering different crises, right? Like for instance, the opiate crisis is huge right now, and even just trying to figure out different testing, um, you know, different testing kits and different reagents, different ways to actually figure out if someone has overdosed on say methamphetamine, right, for instance. Because a lot of the time when we're faced with crises, like right when we have first responders responding to individuals who maybe have OD'd on something, right, we sometimes don't know how to properly remedy that issue, because if you've given them the wrong drug or give them the wrong therapy, then they could potentially overdose and die off from that, right? So just identifying what exactly the issue is, identifying the drug specifically, and high specificity, because we need to be on point about everything, right? Especially with not only with hospitals and that type of screening, but along with rehab centers, right? Because we need to be ensuring those individuals who are trying to rehabilitate, right, that they have the right testing. So one of the biggest, I think one of the coolest ones or more revolutionary ones was we've come up with a metrogeny test recently, which is pretty huge. I don't know if you guys know what metrogeny is. It's actually it's based off of a small opiate from a plant called kratom, which is a Southeast Asian plant. You actually create it. It's like a white powder. You can find it in like any drugstore. Actually, you can buy it anywhere in the United States, but people usually make it in teas. They ingest it. But what kratom does is it's obviously an opioid, right? It'll make you sleepy, and it actually has been proven to cause fatalities in very like even in minimal doses. So it's not something that is safe, right? But it's crazy because it's actually not banned. It's not a Class A drug, right, by the FDA. So that's for instance a crazy like that could be very very monumental in terms of that can kill off people. We need to ensure that hey, we need to test for those types of drugs, as if that's not being screened for, and we don't know what's going on with someone. We have to be able to be at the forefront. So we created a new test for that, which was cool because no one has ever created the metrogeny test. So being on the forefront of that was awesome. Yeah, that's just the one example of one of the tests we do, but we do so many more than that. I just thought that was cool because I've never even even I didn't know what kratom was until someone told me. I'm like, what is that? I've never even heard of this, but yeah, they come up with new drugs every day, like fentanyl, spice, all the street drugs. There's a lot of crazy street drugs out there. People come up with crazy concoctions, man.
All right. Yeah, so that was a good amount of information right over there. So as everyone knows, the field of biotechnology, it's like increasing very rapidly. So where do you think biotechnology is going to end up being, like, say 10, 20 years from now?
Oh, the sky's the limit, man. It's crazy because if you think about it, like we have completely revolutionized vaccinations at this point in time, right? In terms of, although Thermo Fisher, we actually do not directly um, manufacture the vaccines themselves. We help assist in providing the raw material to help create, right? But even then, like this whole new sequencing of viral vector vaccines and the mRNA vaccinations, even these entire processes are revolutionary in terms of now when the next virus hits, the next bacteria, the next strain of anything, we actually have a method to actually manufacture the vaccine at such a rate, right? A faster rate than now. And that is crazy to think about. That's only in vaccinations, right? That's just the tip of the iceberg. When we're thinking about cancer, cancer development, for instance, they've actually utilized similar technology in counteracting cancer cells. Actually, that's actually where they figured out where to utilize this mRNA vaccination technique. It was actually through the NIH. The NIH actually does a lot of cancer therapy and cancer work. So it's actually kind of cool what all of this stuff overlaps, and we're all kind of in conjecture working with each other. So it's just crazy. Like you think one thing about biotechnology, but the moment you enter the sphere, the sky's the limit. Like there's so much hope in ALS. Finding out a cure for schizophrenia, for instance. We barely know anything about the brain, right? In terms of just understanding our neural connections and like even memory. It's just incredible. Yeah, that's just some aspects that I just am fascinated about as well.
Yeah. So while we're on the topic of the future in biotech, someone from the chat had this question. Do you think biotech could end up going green or turning CO2 into oxygen?
Ooh, going green, maybe that'd be cool. Hey man, you guys can be providing the solutions to these very problems. Like that's something we can all try to come up with ourselves. This is something that I didn't think was possible in terms of I didn't think it was possible to come up with a vaccine within 6 to 7 months, yet they did it, right? That is in itself a miracle. Like I can't tell you that's a scientific miracle in itself, because to be able to develop it and have phase 3 trials that are 94% effective is astounding. Like that is astounding because they were actually proven. And if you look at the research and the people who have the side effects were only rashes, if that, maybe like a little bit of flu-like mild symptoms, but the symptoms were nothing. Like it is revolutionary in terms of this is actually going to be more prolific. And yeah, man, CO2 into oxygen. We just got to come think about the right reaction, right? Like let's see, like some sort of photosynthesizing, like because technically with photosynthesis you are creating oxygen. If you could if you genetically engineer some some crazy botanist. I do like botany as well, so this is a solution the botanist can come up with too. I'll be the first one on the team.
Awesome. Okay, so someone has a question regarding your college life. So what biotech program did you attend and how was it? Was it like a difficult program or was it relatively easy?
It was a little bit of both. I think the challenging aspects of it were I went to Santa Barbara. I apologize for not making that clear. So I went to Santa Barbara. The program itself was good. I felt like I was challenged enough, because for me, if I'm not challenged in either the academic sphere, then I'm going to be elsewhere. Because for me, like I need to be challenged. I'm the type of person who is very bored in class. I get bored very easily if I'm not being like on the edge of my seat, tested to the edges of my mind. So for me, I think our organic chemistry courses were very, very challenging. But I really enjoyed it. I think my favorite classes were definitely organic chemistry and biochemistry, mainly because how the understanding of the breaking down of food specifically was something that I think I just explained to you guys as well a little bit about that. But like that's something I was really fascinated by, as well as just organic chemistry was just so cool because you can basically um, come up with so many different types of molecules just from carbon, right? Carbon, hydrogen, and oxygen. Like it's insane. the amount of combinations. You can like, there's so much flexibility, and just the amount of organic, like the reactions themselves were just complicated to learn, and the mechanisms were really excruciating at the time. But I'm grateful, because now I understand a little bit about why they work the way they do, right? Like, it helps to understand the mechanisms, so that when you're in the lab, all of the little, the excruciating work you did to kind of fit that material in your mind, it's all applicable now, right? Like, now all of the, anytime my experiment's going wrong, I'm like, okay, I just need to fix this by, maybe I have to add a little more of a nitrate, right? Because the the nitrate reaction is not happening fast enough, so I have to add some more, maybe, nitric acid, whatever it is, and that helps me, especially with my process. But yeah, I think the biotechnology courses were good in Santa Barbara. They were challenging enough, which is good. I would highly recommend going to a challenging enough program. Specifically for biotechnology, I would highly recommend a couple of different programs. The program that I really was looking into the most was, there was one at Davis, I think, has one. I know in SD, they do a lot of medical research, as well as their School of Medicine has so much going on there. I think they're doing a lot in terms of the biotech sphere as well in San Diego. And USF, School of Medicine, although USF, I would say their graduate programs, once you get to that field, unbeatable, like unbeatable. They do a great, they have great programs up there. And then I think USC also has a lot of online programs, if I'm not mistaken, just because I know a lot of the times, most people have, even now it's so much harder to do things in person. It's basically impossible. And most of these classes are, available. So you can just try them out, see how you like them. If you don't, hey.
Yeah. So besides science classes in your college, 'cause I just want to keep on staying on the college topic. It's really interesting in my opinion. Were there any other classes you had to take, like math class or any other sort of class?
Oh yeah, I took calculus. I had to go. Yeah, definitely took calculus. I love math. Physics, for instance, definitely took physics. That was fun. Yeah, I had to take a lot of different courses. We had our GE requirements, so you had your own subset classes you had to take for graduation, along with your requirements for your scientific degree, right? So those classes were really fun, though. I took like, what did I take? Oh, I took like philosophy classes, and I took a couple of sociology classes, because I also originally wanted to be a doctor. So I took the MCAT afterwards. So I had to take a little bit of sociology, psychology, and I took a lot of other philosophy classes, which were really cool.
Yeah, that's really cool too. So we have another question in the chat, but this one's more about your current work situation. So what kind of tools would you often use in your lab?
So the tools that we use, a lot of instruments. We use like centrifuges. We use like pipettes. We have a lot of large analyzers to help either analyze whatever material we're using. We're identifying like whatever items we're utilizing. We have like large hoods. If I was to show you a picture, I really want, the thing that I need to do next is, I really wish I had time to do this. If I went through our entire warehouse and like took a video of just all the instruments, it would blow you away. It's huge. Like, I don't do it justice by describing it this way. What I would highly recommend is, at some time maybe I could try to get a recording, but if possible, look on the Thermo Fisher website. They might have a picture of the inside of the labs, because they're huge. Like, the warehouses themselves are massive. There are so many instruments in there. I can't even name it. Like, I'm not even doing even remote justice. There's like photospectroscopies, and they have like linear chromatography machines, and TLC, like thin line chromatography machines. They have proton NMR, which is nucleo magnetoresonance machines, which they're like these like $500,000 machines that are just circular. You put a little bit of sample inside of it, and then it'll tell you where exactly the most how do I say, that's too complicated. I'm not going to go there, but it'll tell you like the different carbons that are the most polar and reactive. So yeah, it's cool. And hydrogen specifically. But yes, we have a lot of cool machines like that. If anyone has any other questions, let me know. I do have to go in a little bit. I do apologize about
Okay,
that.
yeah.
I just had to pray like 3-4 minutes, but I'm so happy I was here and able to talk to you all.
All right, yeah, awesome. So I guess that's the end of this workshop. Mr. Sajid Hai, if you have any last words you'd like to give to the audience, now would be the right time.
Absolutely. Um, yeah, thank you for all the questions you have to make a. Yeah, no, you don't have to make a scientific breakthrough technically. Um, the good thing about being a part of such a large company is that you can kind of see where your role is, and I guess in a way. Although I think everyone's own findings and contributions might not be huge in the long run, obviously with like tens of thousands of employees, but it helps to know that your function and your role plays a huge integral part in this massive machine that is kind of churning out the best solutions to countering the pandemic, countering all of these different issues that we have in front of us. And it's just cool. Like it helps me have a good sense of I'm doing a good job in my everyday life and making a difference. As well as, yeah, I do have a little bit of research in my own job. Like we send samples out to different, we send out different, research samples that we're working on that we might not have as an approved product that we're still testing, for instance. But yeah, we have a lot of opportunity definitely in our company. If any of you are interested, we do have a career website as well. Feel free to check it out. If you have any questions about anything on the site, feel free to reach out to me directly. I can give you my email address. It's just my first name dot my last name at thermofisher.com. Actually, I'll actually message it, put in the message right now, just so if you guys have any other questions, you feel free to message me or send me an email. I do answer almost immediately. If I do not answer immediately, I do apologize. I'm a little bit busy some days, but most of the time I am free. But feel free to look out the website. We do post summer volunteer opportunities, so do not fret. I will make sure to keep you guys in the loop if you guys have anything that you wanted to look into, especially in Thermo Fisher Scientific. We do have locations in Sunnyvale, in Pleasanton. We have a location in San Francisco and Fremont, so you don't have to go too far as well.
All right, awesome. So thank you so much for your time. I'm sure everyone loved to hear what you had to say. So yeah, I guess that's the end of this workshop, and we'll all see you at the closing ceremony. So thank you everyone for coming.